Okay, but then before we dive a little bit deeper into immunology and also tumor biology, I would like to also give you a very brief introduction on our company. And the company was founded in 2018 by myself and my co-founders from Yale University. And we are currently headquartered in Cologne, Germany, but with also sites and labs everywhere. We have, for example, two sites in the US, in Connecticut and also in Michigan. And we also have lab in Singapore, as well as quite extensive service and clinical lab in China and in two different cities. And in Germany, we have our service lab that can take on samples from all over Europe. We also have our instrument and reagent development and manufacturing. Of course, we also have our commercial team in Germany, and that can serve our customers all over the world. And so far, we have made some progress in the last almost 8 years after the company was founded. Now, worldwide, we have customers in more than 30 different countries. We have also more than 700 customer publications in good journals, and also more than 5,000 labs worldwide using our technologies. And altogether, we did some counts from our service lab as well as our customers. Our technology have been used to sequence more than 1 billion cells, single cells already. And this number is, of course, also growing. And we are very proud of the fact that our technology can really help people to do better and maybe also faster research. And here are just some customer testimonials from our customers in different countries. And also we are a proud member of the EU projects, Certainty for blood cancer and B2B rare for rare disease, where the single cell multi-omics technology can be used for better treatment and diagnosis. And we are also official partner for the Global Human Cell Atlas Consortium. And here is just like one example of the high-level publications that our customers were generating using our technology. And for those of you who are probably relatively new to the single cell technology field, I would like to give a very brief introduction on how the technology works. And so this is, we are talking about not like the getting one single cell and analyze its transcriptome or genome or proteome. We need to analyze like thousands of cells all at once, but with single cell information, because each cell is different. We have altogether about 3.7 trillion cells in our body, although the genome is more or less the same in normal conditions in each cell, but we do still have different organs, different tissues, and different functions, and those are really coming from the heterogeneity of each individual cells. And to really analyze those cells and get their individual characters, we need to have some new technology to first label each cell. So this is, for example, how Singleron does it. Of course, there are also other technology approaches. So first, we would need to really separate cells we call it partition. And we use a microwire-based chip and also Poisson distribution. So if we load a cell suspension to our chip, for example, this is what we use, a very simple manual chip that Søren also tested already in his lab. And then based on Poisson distribution, the cells will be dispersed. We have 150,000 microwells on this chip. And if we load, for example, only 10,000 cells, then they will be dispersed in such a way. Most of the wells will have no cell, and some of the wells will have one cell, but the chance of like more than two cells or more than two cells coming to the same well in this condition will be very low. So that's how we separate the cells from each other. But the next step is to barcode them, because each cell will need to have a personal ID. And we use barcoding beads to do that. And we have like magnetic barcoding beads, and each bead would have about 10 to the 6th copies of cell barcode oligos with the unique sequence on top of their surface. And but any two beads would have like different barcode sequences. And then we load those beads also onto the same chip. The beads would have a dimension that would prevent more than two beads coming into the same microwell because of the dimension. And so each bead will fall into each well, and then we lyse the cells, and the beads, in addition to the barcode, also have oligo-DT. So the oligo-DT can capture then the poly-A tails of those mRNA inside of each cell. And after that, we capture like all the mRNAs from our cells, and we can do one RT reaction and one library. But now the mRNA coming from each individual cell will acquire this unique barcode from the barcoding beads. That's how we could use bioinformatic tools to determine which information comes from which cell. And yeah, so that's how we could get the single cell level transcriptomics and other omics information from thousands or tens of thousands of cells all at once from each individual sample. And this could really help us to gain much deeper insights into the heterogeneity of any sample and any system. And I assume most people here work either in immunology or tumor biology. As you probably all know, immunology and also cancer, they are like both very complex and very heterogeneous systems. And using such a tool as high throughput single cell sequencing is actually perfect fit for this kind of complex system, because you now can see like the individual characters of those individual cells. And yeah, so we are a company that's very focused in single cell multi-omics solutions. And we also understand, actually, if you have a tumor sample, or if you would like to study, for example, the immune cells inside of a tissue, you have to, sometimes the bottleneck already starts from the beginning in dissociating those tissues to good cell suspensions with good viability and also good account. So that's why we try to provide a whole solution from the beginning to the end, starting with the tissue dissociation. And after that, we also have, as I just mentioned, based on the micro chip, we do have the library prep system to separate and also barcode each individual cell so that we can collect their genetic information. And yeah, so we have, once our libraries can be sequenced on basically any sequencing platform, whether it's Illumina short read or ONT long read, it's compatible. And once we have the data, we have cloud-based data analysis tools, as well as a database that can help researchers to generate more usable information from the raw data they get from the sequencers. And for tissue processing as well as the library preps, we have the simple manual solutions that you can just buy a kit and do it immediately in your lab. We also have automation. So for people who have higher throughput, who would need higher standardization of their workflow. And maybe just like a brief introduction on the tissue dissociation solutions we have. Normally, I don't know how many of you have done tissue dissociation yourself in the lab. Normally, it's could be quite tedious. And if you search online, you see a lot of information, a lot of protocols tailored for different tissue types, and you probably have to do optimizations yourself, and that's a bottleneck we wanted to overcome with our kit. So we do have a tissue dissociation kit as Cell Life that we use that works with a majority of different tissue types from different species, from for example, not only human and mouse, but also some seafoods like fish and other animals, and that also works with different organs. And it works with large tissues coming from animals or coming from, for example, also the resected like surgery samples, but it also works with very tiny biopsies. And yeah, so there have been like hundreds of papers already using our tissue dissociation solutions. And normally, if you do have a very difficult tissue type that you would like to crack, you can always just talk to our tech support colleagues, and they can give you information on whether we have already tested it in our own service lab, because we have service lab in Europe, in Asia, and also in US. We have a lot of also hands-on experience ourselves with different tissue types, which can help you probably to crack the difficult, challenging tissue types more quickly. And we also have the automation, the Python I instrument that can be used to dissociate different tissue types with our Cell Life dissociation buffer. And additionally, this machine can run like 1 to 8 different tissue types all at once, and you can use different protocols in the same run, meaning you can have 8 different tissue types. Each one of them would have different protocol regarding the speed of the this grinding, as well as whether it's clockwise and counterclockwise, and the time of incubation, and to have the flexibility if people work with different tissue types. And this machine also for a molecular biology lab who that might also need to extract DNA and RNA or protein for further analysis. It also works to homogenize different tissues, and so that the extraction can be done. And this also works to extract nuclei, just in case you might be interested in nuclei-based analysis, like either protein analysis or MSA, or maybe single nucleus sequencing in some cases. And as mentioned, the automation has the advantage compared to the manual workflow of the reproducibility, and we get the same high viability as the manual kit. But no matter who the operator is, the data could be kept reproducible. And and also, as I also mentioned, our tissue dissociation solution also works with tiny amount of tissues. So like even with a biopsy, if it's only like about 10.5 nanogram, it also works to get single cells out of them. And so I would skip this part. If you have more questions, of course, we can also discuss. Then I would also come to the core of the technology. we provide, which is the sequencing library construction from different sample types. And so as mentioned, for RNA, it's relatively straightforward. We just use the barcoding beads with oligo-DT to capture all those different mRNAs. And we have different formats and different throughput to capture up to 30,000 cells all at once from each sample. So that would give you, for most of research projects, quite sufficient cell number to account to detect even very rare population of cell types. And here is just to show you how the chip would look under microscope. We have all those small wells with about 40 micron in diameter, and then through the Poisson distribution, those cells can be partitioned, and then their genetic materials barcoded and collected. And yeah, so this is typical data you will get from single cell RNA sequencing, and based on the expression level of all those thousands of different transcripts, you can determine which cell types they are. This is like an example of a whole mouse embryo. You can detect different epithelial, endothelial cells, and also there are different subtypes. You can also detect immune cells. And for blood, for example, because we use Poisson distribution and also just like let gravity do the job, since the cells will just fall into the microwell very gently, we don't disrupt the fragile cells like some other technology might do. So we preserve, for example, also quite fragile cells in immune system, like the neutrophils, basophils, and platelets, and so on. And this is just one example. And yeah, again, this can be done manually to get a quick start. We also have a machine matrix new that can be used to run the process in a more automated way. And for labs that are doing a lot of samples all at once, we also have the high throughput tensor that can go from cell suspension to sequencing ready libraries all on one system for up to 16 different samples. And this technology has been used in also a lot of publications. For example, a recent publication from a lab in Switzerland used the single cell GEGscope RNA sequencing kit from us to look at the CD8 T cells. This was published in Nature early this year or late last year. I cannot remember exactly, but they could use this technology to look at the subtypes of CD8 positive T cells in the mouse with type 2 cytokine treatment. And then they could use the method to really determine not only like the change of different CD8 positive T cells proportions before and after the cytokine treatment, but also the critical immune suppression and immune activating gene expression before and after the treatment. That's really the power of single cell sequencing, because, for example, using FACS, as they also used in this study, you can only look at the cell population. You don't really directly have the information regarding their internal signaling pathways and the expression of genes that you cannot directly with the FACS analysis. But of course, there are a lot of other things other than mRNA in a cell. So that's also why we also have multi-omic single cell products that can detect, in addition to the mRNA, also other analytes inside of a cell. Maybe more important for both immunologists and tumor biologists would be the immune receptors inside of sample, whether it's for immunology study or whether it's to look at the clonality of tumor-specific T cells or B cells. So we have a circle product that can detect mRNA as well as TCR and BCR sequences from the same immune cell. And we also have a focal scope product that I don't have time to talk much today, that can detect both mRNA and also like mutations as well as fusions and also splicing isoforms from the same single cell. And we also have the promoscope. As some of you might know, the cell surface glycosylation in the immune cells is related, is different at different development stages, and is also related to the activation stages of those immune cells. We also have a kit that can be used to detect both mRNA expression as well as the surface glycosylation level of immune cells and of course of other cells as well. And a Datascope kit that can be used to detect both micro mRNA and also the ratio of nascent RNA to study the regulation of the transcriptomes inside of a cell. And yeah, just maybe going quickly to the circle for if you are interested in our products. We have the brochure here, and also Ritika and I will be here to answer any questions. And I just want to show also very quickly this one customer publication on Cell magazine this year. They use our circle kit to detect not only the T cells in the microenvironment of ovary cancer patients. They also could use the methods to using the T cell sequence, T cell receptor sequence. They can track the tumor-specific T cell clones and also how those T cell clones evolve in the patients, also in different, especially in patients with different drug treatment responses. Yeah, so I suppose we are getting probably, then the time here is wrong. I was thinking that I'm running out of time. Then looks like we still have maybe 10 minutes or 15 minutes. Okay, thanks. Yeah, looks like the time here is probably not, I misunderstood the time on the screen. Let's put it this way. Then I have still have 10 more minutes that I can, for example, also give a brief introduction on the focal scope kit that can be used in a quite either standard or customized way to, for example, if you have some tumor cells, right? And you are probably familiar with the driver mutations. But those driver mutations in the tumor cells, they definitely are not expressed in each and every tumor cell inside of a tumor. Some tumor cells will have those driver mutations expressed, some other tumor cells will not. So which cells will express those mutations and which cells will not, and also the expression of mutations, what can it do to other pathways in that tumor cell? It's, I think a lot of questions can be still asked and also answered. And to address that, we have the focal scope kit that can capture the mRNA from the single cells, but at the same time, we can also design different probes on the cell barcoding beads so that it can also with, if you have certain driver mutations, for example, you are interested in our isoform or fusion gene, you are interested, we can capture those together with mRNA so that you can have additional layer of information. And for example, to understand why a driver mutation is a driver. And we have ready to use panels already for blood cancer, for lung cancer, and also for the immune cell mutations through aging, the clonal hematopoiesis mutation panel. And since EBV is also quite important in immune cell function, as well as in quite some different tumor types, starting from blood cancer, but also other cancer types. We also have a ready-to-use EBV panel that can detect virus, as well as mRNA from the single cells. And here is one example on how the EBV positive and EBV negative cells can be divided. And here, if you look at the overlay, you can see that, for example, the Raji cells, I think they are EBV positive, and you can detect also their EBV reads with the focal scope capture method. And this was also already used to study the chronic active Epstein-Barr virus disease, to see, like, because we could detect both mRNA and also EBV reads from single cells. So the method was used to really track how this disease evolved from the stem cell origins, and how, like, the percentage is in different immune cell types. And yeah. And I also briefly mentioned that we have the PromoScope kit, which can be used to, so how we do that is we have a modified oligo that also has a poly A, and this modified oligo can be conjugated to the glycan groups on top, on the surface of a cell. So with enzyme that we produce ourselves. And with this conjugation, each cell will, if they have a lot of glycans on the cell surface, a lot of oligos, these specific oligos can bind to them. And then we do the normal partition as well as capture, as we normally do with our single cell mRNA sequencing. And now the barcoding beads can not only capture the mRNA inside of cells, it will also capture those this modified oligo on the surface of cells if the cells have glycan on their surface. I think they will all have, but the more glycan groups they have, the more this specific oligo will be there. Then in the sequencing, we look for this specific oligo sequence. And with the number of the specific oligo sequence we can detect, we can discover, or we can determine what's the glycosylation level of each individual cell. And here is also one example for example, I think this is mouse bone marrow. And as you can see here, like with the mRNA single cell information, we can do cell annotation. We know which cell types they are based on their specific markers. And then we count this very specific glycan tag to also determine what's the glycosylation level of each individual cell. And this method was also used in this one study that was published in a chemistry journal, actually, and where it was also shown in tumor immunology, tumor biology, actually, the level of the glycosylation in the CD8 positive cells also changes throughout the activation in the tumor process. And the Dynarscope, would probably just skip. Otherwise, but we have all the information either on our website or in our brochure. And of course, you can also ask us if you are interested in how like each individual RNA is regulated, like how quickly they are synthesized. This method can be used to really look at what's the exact percentage of the RNA you detect in each cell is nascent, newly synthesized RNA. I would just like to also mention in the last slides that we have all those kits for tissue dissociation, for library prep of different analytes in the single cell so that they can be sequenced, their information can be disclosed. But then you get a lot of data and that you need to generate insights of. And of course, you have experts like Søren here who can help you, but sometimes he might also get very busy and he might also need some help. And then we have a cloud-based data analysis that can at least take the routine work of data analysis away from a skilled bioinformaticians so that they can focus on more important biological questions and biological analysis. So we have Sally Lan's cloud-based data analysis pipeline. And once you have your single cell data, you don't need to be a professional bioinformatician. You don't need to know how to code our program. You can just upload the data onto our cloud. And then it's like quite easy to use self-explanatory interface that you can upload your data. You can do basic analysis, routine analysis to get all those different cell types. The annotation is also automated. And then you can also compare different types, different samples and look at their pathway analysis, which pathways are activated or downregulated. And yeah, and I think that's about, we also have the database, of course, but if you have the Sally lens, this can be also connected to a database that can generate further insights. And with that, I would like to thank everyone for your attention and also welcome any questions you might have and also comments, of course, feedback. Yeah. Okay. So there are some online questions. Maybe I can, should I go through, answer the online questions first? Yeah, so which species can Singleron platform work with? Because we use this oligo-DT method to capture mRNA. So eukaryotic species, we have theoretically, it can all be done with our platform. In reality, we have our customers have been working with all kinds of different animals and also all kinds of plants. And additionally, we also have a yeast kit that can also be used to work with yeast cells because it can, the specialty of that is lysis buffer that can lyse also the yeast cell wall. As you know, yeast is also getting to be a very well-known model system to study a lot of pathways. Yeah, so as long as there is poly A tail, or there is a sequence that we can design probes to capture, theoretically, we can analyze them. And my lab has not yet done single cell sequencing. Do you provide training, consultation? Yeah, that's also a very good question. We do have like a very strong tech support team in Germany, like our PhD scientists experience in single cell sequencing. So they can, if you have any question, just feel free to write to us. Our email is info@singleron.bio. you can fill a form online on our website and just like ask for a consultation session with our tech support scientists, and then they can have a meeting with you to discuss what would be the best solutions for you to perform a single cell sequencing experiment. And the next one, how does the customized focus scope work? Can we pick any mutations? Theoretically, yes. If you are interested in detecting different mutations, you can pick any mutations, and then our workflow is first, our scientists will evaluate the mutations, and depending on where they are, they can decide how many probes we will need to capture them. And we haven't, for all the customization projects, we haven't encountered like a situation where we say, okay, this is a very difficult mutation, we cannot design a probe to capture it. And so once they have evaluated it and designed probes in silico, it would take normally 6 to 8 weeks for the production, as well as validation, because for each batch, we will need also real single cell experiments to QC them to make sure it works also in real samples. We normally suggest to detect less than 10 different mutation regions, because if we put too many probes on the top of our beads, the capture rate for the mRNA might be decreased. So that's the only limitation. But we do also have customers who say they don't care if they capture a little bit less genes in the mRNA parts, that they want to capture like 20 different mutations. That also works. Yeah. Okay, so I think we are good with online questions. If we have any questions, this is for online. Okay. I have 2 questions actually. Okay. Thanks.
I'm giving a lot of presentation diagnosis, and we biopsy between 5 and 10 cells in the blastocyst. Could you tell me if that technology could be applied to the blastocyst cells if there are only 5 to 10 cells to detect some mutation if the famous test? And also, is it applicable in the fetal DNA that is still free from the pregnant clone?
Yeah, it's a very good question. So the technology I just showed that's based on our microwell chip doesn't work with 5 to 10 cells because you always lose some cells after you load it. And also if you have only 5 to 10, it will be very difficult to capture all of them. But we do get this kind of also requests and desire to study like selected single cells. And we do have a product I didn't have time to talk about that's called AccuraScope. It's a well-based technology where you can seed, like it's 96 or 384 well-based technology. You can just seed your single cells 1 by 1 to each well. And after that, we lyse the cells in the well. We also barcode them in the well too, so that we can process, for example, all your 10 cells all at once for both DNA and RNA. And this can also ensure the genetic material is captured more sufficiently, because with 1 cell, the amount is like only 6 picogram of RNA and about 10 to 20 picogram of 6 of DNA and 10 to 20 of RNA. That would be very difficult. But if you put 10 together, first the cost would be lower, and then also the DNA and RNA amount would be higher. It would also ensure better capture of all those. If you are interested, yeah, you can just write to us and we can send you more information. this is also for you to get both DNA and RNA sequencing information from those few cells. And for the fetal circulating DNA, no, so our technology doesn't work directly with circulating cell-free DNA, but theoretically, you can just like, extract them with, for example, the cell-free DNA or RNA kit, and after that, you can use our any library prep kit to generate library from the cell-free DNA, and yeah. So that's actually a very easy, I feel, relatively easy problem to solve. You can use, we have a standalone library kit, and yeah, that can also be used for that. Yeah, but thanks for the questions. And any other questions? Okay, if not yet, I wouldn't block Soren from showing his exciting data. And we also have the, oh, okay, please.
Congratulations, your technology is great.
Yeah, thanks.
About during this process of single cell producing. Do you have results from these regimes of single cell producing? Is it affected gene expression? If you have data, please share with us this information, because it's interesting. In single cell processing, is it affected RNA expression?
Yeah, so mostly I think we are looking at RNA expression in those single cells.
Means that the process of producing the single cells does not affect expression.
Yeah, actually, I think this is a very excellent question. Yeah, exactly. And not only for single cell RNA sequencing, but even for bulk RNA sequencing, there has been quite some studies showing actually how you process your sample will affect the transcriptome more or less. For example, even for collecting samples, if you collect a sample from human patients, like how quickly, for example, either you freeze the sample or further process the sample for RNA extraction would already matter to how the transcriptome would look exactly. And also the extraction methods, RNA extraction methods, different companies' methods would also produce some difference, as well as the library prep method. And also for single cell, of course, there could always be like the dissociation of the tissues to cells could also potentially produce a certain change in the cell transcriptome. Like I really think as soon as you get a biological sample from that biological body, whether it's human or animal or plants, it will change. But then the trick here is that if you do like one experiment, if you want to compare different samples, then you should use the same protocol from like collecting and also sample processing, either it's dissociation or extraction and library prep, so that you remove, you have those variations caused by, but they will be normalized. Exactly, that's what normally people suggest for RNA-related studies. But that's a very excellent question. It's really like what people try to solve in these fields, but it's not always so easy.
Yeah. Expression, and I know that multi-carrying is very sensitive and change the lifetime of the RNA is very short. And this whole manipulation is enough time to degrade.
That's true. But maybe for tissue, I didn't have time to mention, but we do have a tissue preservation solution that can preserve. So once you have a fresh tissue, you can put the tissue in this preservation solution. And for up to 72 hours, the cells will kept alive. And we also compare that to freshly extracted tissues. Actually, the transcriptome profile is also quite similar to the fresh tissues. But you can try to reduce the effect, but totally eliminate, I think it's probably not possible. You just have to maybe use the same technology for all samples in one study group. Yeah, sure. Oh.
Hello.
Hi.
I'm Jordan from Medical University of Sofia. The previous question made me curious. What's your experience if you combine bioinformatically the single cell data? Does it reconstitute the boxy result more or less?
It does in certain way. I think that's why like you can really also pull like single cell RNA sequencing data from like from a sample and reconstitute the bulk RNA sequencing data. So that's what people are also doing. But then you need to keep in mind, maybe like if you have a big chunk of tissue, you have data from maybe, I don't know, 1 million cells. And with a single cell, you have data probably only from 10,000, 20,000 cells. So there might be some difference considering even like different positions of a tissue could have different cell populations. And but what's probably also interesting if you want to know is you can also even use deconvolution to take bulk RNA sequencing data and do pseudo RNA single cell, get pseudo RNA single cell RNA results. Yeah.
Yes. So if it's the same number of cells, more or less, both sequencing of the sensitivity and the number of transcripts should be similar.
I would say it's not identical because you do capture like at single cell level, normally we capture like thousands of genes all at once. And normally if we have like 10,000 cells, we do get total RNA more than 20,000 genes, which is similar to what bulk has. But on the other side, also depends on the RNA content, transcript content, as well as the methods, extraction methods, and library prep method you use. I think there will be some difference in some genes.
Yeah, thanks.
Yeah. Okay, but thank you very much. It's also very good question. I think in a big picture, they are similar, but maybe if you zoom in at each individual genes or RNA, there might always be some difference. Yeah. Okay. We can also discuss further, of course. Yeah, later. And but thank you very much for all your interest. And yeah, it's also
Thanks. Talk.